Our final topic for the semester is cellular respiration. Cellular respiration will be a little bit easier since we've already covered photosynthesis. A lot of the same kinds of themes and patterns that we've seen in photosynthesis we'll also see in cellular respiration. I'd like to start out by doing some comparisons between three different processes that you are familiar with to help us understand how similar they are to cellular respiration. So I wanted to talk first of all a little bit about how your car works in the combustion of gasoline. I want to think a little bit about burning a candle, and we'll be able to compare that to cellular respiration. So let's look first at the fuel. We know that when you fuel up, you put gasoline in your car, and gasoline has a lot of carbon, hydrogen, and oxygen. It's very similar actually in structure to glucose, which we know is going to be the fuel for cellular respiration. And if we want to, we can remind ourselves that C6H12O6. When you burn a candle, what you're actually using as your fuel is a type of wax. And this wax is oftentimes a paraffin wax, so these are usually what candles are made of, but if you have a soy wax candle, then it's just a different source of that wax. That wax has to have energy in the bonds holding the molecules together so that you can then burn it and actually get light energy out of that. Let's think a little bit about whether or not oxygen is required. We know that in your car you have to have an oxygen sensor so that you have the right amount of oxygen so that you can ignite and burn that gasoline. I hope you know that when you're burning a candle, you also have to have plenty of oxygen as well. Even if you think about a campfire that you might have, oftentimes to get it started, you'll blow on it, and what you're doing is actually increasing the amount of oxygen around it. And of course, we know that living organisms, vast majority of them, not all of them, have to have oxygen available to be able to break down their glucose. We'll look at a couple scenarios in humans later on in this unit, talking about how we can actually utilize energy and glucose without oxygen for a very short period of time in certain cells in the body. And actually some organisms can live completely without oxygen, and they have some different strategies in this process. But by and large, our discussion as we move forward right now is going to be assuming that oxygen is available so that glucose can be broken down in the most efficient way possible. When we think about what happens with the energy that is going to be produced, we want to know if it does work. Well, of course, in your car, when you break down that gasoline into energy, moving that chemical energy into kinetic energy, the energy is used for work, and that's to make the wheels on the car go around. I hope it's also very clear in cellular respiration that the whole point of breaking down glucose and extracting the energy from the bonds of that molecule is to do work, chemical work, transport work, mechanical work in the cell. And I think we've talked about that back when we looked at energy. When we think about burning a candle, we're not really doing any work per se. Finally, we want to know what the products are, the end products of each of these three scenarios. I hope we remember from our equation of cellular respiration that we will have an output of carbon dioxide and also of water. What's interesting about this process, though, is how similar it is to the combustion of gasoline, because when you break down gasoline in your car, you also produce carbon dioxide. That's what's coming out of your tailpipe, and you'll also produce water. So the products for our car burning gasoline are exactly the same as the process of how cells use glucose and produce CO2 and water. And interestingly enough, when you burn paraffin wax or any type of wax, some carbon dioxide is produced and some water is produced as well. So I hope it's easy to see how similar these are to one another when we think about whether we're burning gasoline, burning a candle wax, or whether we are looking at cellular respiration inside of the cell. Therefore, I hope that it's easy for us to remember our general equation for cellular respiration. Let's write that out again here. C6H12O6, and oxygen's required for that, and then we're going to break that down into carbon dioxide and water and also get the energy out of that as well. It's important that we remember what's being oxidized and what's being reduced. So let's remember our terminology. Oxidation is loss, so we want to find our reactant that is losing electrons. And remember, we're always looking at the hydrogens to find out where those are. So based on our equation, we can see that our hydrogens are going to be in glucose, And if we remove those hydrogens lost, we are oxidizing glucose to CO2. So that's what's being oxidized in our chemical reaction. And then we also need to know what's being reduced, what is going to gain the electrons. And I hope we can see that oxygen is going to gain those hydrogens, and so the oxygen is being reduced to water. So that's what's happening in the course of cellular respiration. There's an oxidation, there's a reduction, and we can see which are oxidized and which are reduced. Before we get into talking about some of our details, it's important that we remind ourselves of where we are inside of the cell. I hope inside of cells we remember the mitochondria. And earlier on in our semester we said mitochondria have the responsibility of making energy for the cell. I hope that we can restate that and say it a little bit better, which is that it's the job of mitochondria to transform energy in one form to energy in another form for the cellular's use. So on our big picture, what we need to do is give C6H12O6 to our mitochondria, and mitochondria are going to transform or convert that into another type of energy that can actually be used in the cell, and that's our ATP. Let's take a look at the structure of a mitochondrion. You don't need to know all of the structures listed here. I just wanted us to notice a couple of things. First of all, you'll notice that there are two membranes. So first of all, there is an outer membrane of our mitochondria, and then you'll also notice that there is an inner membrane of the mitochondria. So that inner membrane is highly folded. So here's this membrane, and I'm outlining it here in purple. This is the inner membrane, and you can see that it has all of these folds in it. And it's important that we understand why. Anytime we have a lot of folds in an area, what that does for us is that it increases the surface area. It means there's more membrane packed into a smaller area. And if we have more surface area, then we can carry out more chemical reactions, which means that the cell can make more ATP. So this inner membrane is going to turn out to be really important to us. When you look at all of the folds that are formed, the folds themselves actually have a name, and those folds are termed cristae. So cristae are going to be the folds of the inner membrane. Finally, by having an inner membrane and an outer membrane, we end up with some different spaces that are present. So I want to make sure that we're clear about where these different spaces are. First of all, there is a space between the inner membrane and the outer membrane, and that's called the intermembrane space. So the space that's going to be between our inner and outer membrane, that's called our intermembrane space. And then we also can see that there's going to be space on the inside of our inner membrane, the very centermost portion of our mitochondria, and that is going to be the matrix. So matrix is all of the stuff on the inside of the inner membrane. The reason I mention these spaces is because they become very important when we're looking at the process of cellular respiration. And so you can see that the inner membrane really divides these two spaces. It divides the inside-most matrix from the intermembrane space, which is between that inner and outer membrane. Later in our discussion, we're going to zoom in on this inner membrane, and we want to keep close tabs on where the intermembrane space is and where the matrix is, because there are some important things that are happening across this inner membrane. So that's why I wanted to be sure that we mentioned our structure of our mitochondrion. What I'd like to do is a brief big picture overview of cellular respiration, and I'd like you to follow along in your course manual as we go. And then as we move forward, we're going to be zooming in on each of these pieces. Like photosynthesis or meiosis or DNA replication, going through cellular respiration does require a couple of times through for you to get it. And so we're going to go through it one time now, big picture, and then we'll dig in and hopefully you'll have it down. Cellular respiration happens in 3 or 4 steps, depending on how you term some of these steps. Some textbooks will say that it happens in 3 steps, some in 4 steps. I'm going to put it in 4 steps here and give you some alternate names just so that you're clear when you're maybe watching different videos online or reading your textbook. So big picture, we need to understand where these 4 steps are. And the first step that we'll talk about here in just a minute is going to be following along in this arrow, and this is the step called glycolysis. Glycolysis is our first step. And glycolysis is actually going to happen in the cytoplasm. Glycolysis is not going to happen in the mitochondrion, so we're not actually in the mitochondrion yet. The second step that we'll identify some inputs and outputs for in just a moment is what we call the grooming step, and that's along this curvy arrow here. Sometimes this grooming step is also referred to as the transition step. So transition step or grooming step. Our third step is the citric acid cycle. It's important we know that this is different from the Calvin cycle that we saw over in photosynthesis. So here in cellular respiration we have the citric acid cycle. Photosynthesis had the Calvin cycle. And then finally at the end we have what's called an electron transport system, or an electron transport chain. I hope that sounds familiar to you. We also had an electron transport system in the light reactions and photosynthesis, and it actually works here really similarly to how it did there. So when we get to that part, I think you'll recognize how we can actually use an electron transport system to make ATP. So these are our 4 steps, and what we need to know right now for each of them is what is our input and what is our output for each of these steps. So let's look at glycolysis in a little bit more detail to find out what our inputs are and what our outputs are going to be. So we have glycolysis here, and this glycolysis is occurring in the cytoplasm, not in our mitochondrion. Our input into glycolysis then is going to be glucose, which we remember is C6H12O6. We are going to keep track of some of our carbons and hydrogens as we go, so we'll make sure that we tally these up from time to time as we go. So right now we've got 6 atoms of carbon in our glucose and 12 of hydrogen. Throughout the process of glycolysis, we really have 3 outputs we need to know. First off, there is some energy actually made in glycolysis, and that is 2 molecules of ATP. Remember, the whole point of this entire process of cellular respiration is to produce ATP. Now, 2 ATP is not a lot of ATP, but it's a little bit of ATP. And later, as we're tallying that up, we'll want to recognize that 2 ATP molecules get made in this very first step. The next thing that's going to be produced in glycolysis are 2 molecules of NADH. That may sound familiar to us as well. If you remember back in photosynthesis, we had NADP plus was an empty electron carrier, and NADPH was an electron carrier that was full. Here in cellular respiration, we also have electron carriers. However, they don't have the P in it. It's a slightly different molecule. So anytime we have NADH, that NADH, and I'm just going to write this off to the side, NADH is going to be a full electron carrier here in cellular respiration. So it really serves the same purpose. It's going to move electrons from one location to another. Remember that we're moving electrons in the form of hydrogen. So one of the things that's happening in glucose is that we're starting to take some of the hydrogens that are here and we're shaving them off, breaking them apart from the carbon and the oxygen and loading them up into our carrier. And at this point we have 2 NADH molecules. The final output of glycolysis that we need to know is something called pyruvic acid. If you could imagine that we had a glucose molecule and we've shaved off some of the hydrogens and we just snap the whole rest of the thing in half, that's what a pyruvic acid is. So if we take that glucose and break it in half, then we're going to have 2 of these. So for 1 molecule of glucose, we will have 2 pyruvic acids that are going to be formed, in addition to the 2 ATP and the 2 NADHs. From this point going forward, we actually have a couple of diversions, depending on whether or not oxygen is present. So if we have a scenario where oxygen is not present, because we haven't used any oxygen yet, so if there's no oxygen, then we are going to see pyruvic acid go down this road. This is not a road we're going to talk about right now. It's something we're going to pick up with after we finish going through the rest of our aerobic process, meaning that we do have oxygen present. If there is oxygen present, then we're going to go down a different road, and that road is this direction here through our curvy arrow into our grooming step or our transition step. And that's the piece that we're going to look at next. So to recap glycolysis, we start with glucose. We produce 2 ATP and 2 NADHs. We are left with 2 pyruvic acids, and all of this is going to take place in the cytoplasm. Let's move on and talk about our next step, the grooming step or the transition step. The input into this grooming step or transition step are going to be the outputs of glycolysis, which are our 2 pyruvic acids. So 2 pyruvic acid, and we know that we will proceed down through this step here only if there's going to be oxygen present. Now we're not using oxygen yet, we will later in this process. But pyruvic acid is only allowed to go into this next step if there is oxygen available at a later step. We're going to have a couple of outputs of our grooming step as we move forward. And one of those first outputs is going to be 2 molecules of carbon dioxide. If we think back to our original glucose, C6H12O6, that then got broken into 2 pyruvic acids, this is the first time we've seen any exit of carbon. So we had initially 6 carbons in our glucose, and now we're seeing that 2 of these carbons are removed and they are making their way out of the cell as carbon dioxide. This is that waste material that we've talked about. We're also removing some hydrogens. Remember, when we think about oxidation and reduction, that's what we're moving around, are these electrons in the forms of these hydrogens. So like we had in glycolysis, we're also going to remove some of the hydrogens, the electrons, and we're going to load up 2 more electron carriers. So now our pyruvic acid is also losing some of those hydrogens, and we have 2 NADHs that are released. At the end of the grooming step then, if we follow our arrow here to this box, the end product of our transition or our grooming step is something called acetyl, A-C-E-T-Y-L, CoA, and there are 2 of them. Because we put in 2 pyruvic acid, we had to groom 1 pyruvic acid into an acetyl-CoA, and then we had to groom the other pyruvic acid into an acetyl-CoA. And so this acetyl-CoA is the raw material that's at the end of our grooming step. And it's this acetyl-CoA that's going to move into the citric acid cycle, which is our 3rd step. The citric acid cycle is a cycle that is going to turn, and what you'll need to do is to think about what goes into the cycle and what happens for every turn of the cycle. I kind of think of a gumball machine when I'm looking at the citric acid cycle, because you put in your coin and then you crank the knob and then something comes out, hopefully a gumball or a handful of M&Ms or whatever it is that you're buying. And so that's what we need to examine at this cycle. We know that the input of our citric acid cycle then are going to be 2 acetyl-CoAs. And the acetyl-CoA, because there are 2 of them, one will go in the cycle and we'll crank it, and then the second one goes in the cycle and then we'll crank it. And so you could imagine if you had 2 quarters that you were going to put in a gumball machine, you put in one, you crank the handle, you get something out, and then you do the same thing with the other quarter. So the citric acid cycle is going to have some outputs that I would like to summarize. But just to be clear, we're going to have 2 turns per glucose. If we start out with 1 molecule of glucose, then this citric acid cycle has to turn twice. Let's list our outputs for this citric acid cycle. First of all, there are 2 carbon dioxide molecules that are going to be released for every turn of the cycle. So if we multiply that by 2, because we want to calculate this per glucose molecule, there are now a total of 4 carbon dioxide molecules coming out of our citric acid cycle. This is important because we can now account for all of our carbon in our original glucose. Remember, our original glucose had 6 carbons. Here we can see that 4 of them have been released, and the other 2 had been released in our grooming step over here. So at this point, all of the carbon that was in the original glucose molecule has been released as carbon dioxide. I want to make sure that you understand what this means for you. You eat glucose in your food, and that means that all of the carbon in the glucose that you eat is leaving your body as carbon dioxide. So that's an important thing for us to know about this. For every turn of the cycle, there's also going to be one ATP molecule made. So if we turn the cycle twice, that means we have an output total per glucose of 2 ATP molecules. Again, this is not a lot of ATP, but it's some ATP, and the cell can use this to do work. We also have to account for all of the remaining hydrogens that were not shaved off over here in glycolysis or that were removed in our grooming step. And so in the citric acid cycle, for each turn, there are 3 NADHs. So for 2 cycles, that means we have 6 NADHs. And then there's actually another electron carrier in cellular respiration that we've never talked about, but it has the exact same job as NADH, and that is something called FADH2. And we have one of those produced per cycle times 2, so there's going to be 2 FADH2s produced for 2 turns of the cycle. So the outputs of our citric acid cycle then are 2 carbon dioxide, are a total of 4 carbon dioxide, 2 ATP molecules, 6 NADHs, and 2 FADH2s. At this point in our cycle, we actually have completely oxidized our glucose molecule. There's nothing left of the glucose molecule. However, we're really not done with the process of cellular respiration. So we need to now go to our final step, which is going to be our electron transport system or our electron transport chain, to see how we can deal with all of these electron carriers and use those electron carriers to help us generate a lot of ATP through this process of the electron transport system. The fourth and final step of cellular respiration is the electron transport system or the electron transport chain, which should look very similar to what we talked about in photosynthesis in the light reactions. The inputs for our electron transport system are going to be our NADHs, NADH, and our FADH2s. These are the only inputs into this. These, if you remember, are electron carriers. They've been picking up electrons, hydrogens, from our original glucose molecule all the way through glycolysis, the grooming step, the citric acid cycle. And all of those NADHs and all of the FADH2s now are going to go over to our electron transport chain. Our original glucose no longer exists. However, we have essentially harnessed the energy that was in the bonds of glucose into these 2 molecules, NADH and FADH2. Let's be sure that we can count up exactly how many that we have. So let's start with our NADHs. We have 2 coming out of the process of glycolysis, 2 coming out of our grooming step, and we have 6 coming out of the steps of the citric acid cycle. So we actually have a total of 10 NADHs that are going to enter into our electron transport system. Of our FADH2s, there was only one place where those were going to be made, and those FADH2s were made in the citric acid cycle, and there are only 2 of those. What's going to happen in our electron transport system is that our NADHs are going to be dropping off their electrons, and so we know as an electron carrier, the NADHs are going to drop off their electrons. And NADH drops off at one point, and FADHs just drop off their electrons in a slightly different location, and that's why we have a separate box for it. Once NADH has dropped off its electrons and FADH2 has dropped off its electrons, we just need to mention what's going to happen to our electron carriers. When NADH has dropped off its electrons, then the empty form of that electron carrier is NAD+. And that is going to go back to glycolysis or the grooming step or the citric acid cycle, now empty, to go pick up more electrons and bring back more NADHs. So just as we talked about electrons being shuttled back and forth in photosynthesis, it's the same idea here. FAD, when it is empty, is going to be in the form of FAD. There's no plus there. And so FAD is going to go back to the citric acid cycle to pick up more electrons to become a full electron carrier and then, of course, bring them back here to the electron transport chain. So it's doing the exact same thing. So we do want to make sure that we account for those carriers. As our electrons fall down our electron transport chain, we know that they are changing an energy state. They're moving what we can think of from high energy state to low energy state. And if you remember in photosynthesis with an electron transport system, we can use that to pump hydrogen ions across a membrane. When we pump hydrogen ions across a membrane, we created an area of high concentration gradient and an area of a low concentration gradient. And in creating those gradients, we were then able to allow for hydrogen ions to move through ATP synthase and generate a whole lot of ATP. Well, we're going to go through the details of that here a little bit later. I want us to realize that the big picture of what's coming out of all of this electron transport chain is going to be ATP. And as these electrons fall and we capture the energy from our hydrogen ions passing through ATP synthase, we're making a bunch of ATP. So in this figure, that's what I would like us to look at here. This is no small amount of ATP. There's a lot of ATP made, and it is about 28 ATPs in this process. This is where you make the vast majority of your ATP. Looking back, we made a little ATP in glycolysis, about 2. We didn't make any in the grooming step. We made 2 in 2 turns of our citric acid cycle. But that would not be enough ATP to be able to power a living organism, most living organisms. We find that a lot more ATP is necessary, and so it's this electron transport chain generating all this ATP through that enzyme ATP synthase that produces the vast majority of ATP. One last thing that we need to address that's at the bottom of our electron transport chain is one of our inputs and one of our outputs that we haven't talked about at all. We've seen glucose come in, and we've also talked about one of our outputs, carbon dioxide. But so far, we haven't seen oxygen being used anywhere directly, and it's actually here at the end of our electron transport system where oxygen is going to be used, and this is the only place that oxygen is used. The role that oxygen has here at the end of our electron transport system is to catch those electrons that are falling down the electron transport chain from a high energy state to a lower energy state. And so here we say that oxygen is the final electron acceptor. It is here to accept the electron at the bottom of our electron transport chain. And when it does that, when oxygen is going to receive or gain those electrons in the form of hydrogen, I hope you can see that we're going to form water, another one of our outputs. Now, I haven't balanced our equation here. We realize that we have to have half of an oxygen molecule to get 2 hydrogens to make a water, but I just wanted to stay consistent with how we've written these molecules in the past. I want to make sure we really understand the significance of this. The only reason that you need and require oxygen in your body is for one purpose, and this is it. This is the only place that you use oxygen in your cells. All of your cells need oxygen to be able to carry out this process, but the only role that oxygen has is to be the final electron acceptor at the bottom of the electron transport chain, and therefore producing water. I want us to see why this is so important. So let's actually go back to our larger picture so that we can understand why having oxygen here affects the whole system overall, even though it's only used in one place. If oxygen is used here as our final electron acceptor to capture the electrons and therefore make water, what would happen if we didn't have oxygen? Well, let's just play out the scenario so that we can understand this. If we don't have oxygen, then these electrons that are here are no longer going to be able to be passed down the electron transport chain. So they're going to come to a screeching halt. If that happens, then you have these 10 NADHs here that are not going to be able to drop off their electrons. which means that you can't empty out your NAD plus to go back to your citric acid cycle or your grooming step or glycolysis and pick up more. This would also be true of our two FADHs. If you can't drop off your electrons, then we can't recycle our FADs to go back to our citric acid cycle to pick up some more. So essentially, these things are not available. If you don't have your electron carriers, then you would see the citric acid cycle also coming to a halt because there's nothing to grab a hold of those electrons. We would actually see that if that stops, then the grooming step is also going to stop. So you can imagine a factory where at the very end of the process, you have to have people packaging what's coming off of the conveyor belt. And if for some reason they stop and everything starts to back up in our factory, then we're going to start to shut things down as we go, and none of these steps are going to be able to work. This is going to be true of a backup. Our electron transport system will stop, the citric acid cycle will stop, and the grooming step will stop. However, right here, we do see an alternate option. We said earlier that if there's no oxygen, then there's a different pathway that cells can go down from the end of glycolysis. And that's something that we'll address later, but I did want us to see that in general, without oxygen, glycolysis can continue in some cells under some circumstances if there is an alternate pathway available if there's no oxygen. This is not available to all cells all the time, and we'll look at that a little bit later. I hope that going through this big picture is helpful for us. We're going to be going through this process again in more detail. So in our next question set, we'll go through the details of glycolysis, the transition or grooming step, the citric acid cycle, and finally looking again at an electron transport system to see how we can harness or capture the energy of falling electrons into making ATP.